So I'm here today to present you our ISITE project in wine chemistry. The main coordinator of this project is Professor Régis GOUGEON, which excused but could not be here today. And me and Professor Régis GOUGEON was supervised the PhD fellow Sofia Taktalidou, who is here for 3 years, to study the oxidative stability of wines. That concerns especially white wines and where the sulfites are implicated. So this project is in collaboration with two main research teams, one in Munich with Professor Philippe Schmidt-Copeland, where we integrated the ultra-high resolution mass spectrometry, and of course with the ICMUB laboratory from the University of Burgundy and the University of Crete from the NMR analysis. So we're going to talk about wine oxidative stability, and of course, wine oxidative stability is related to the wine quality. So wine quality is a big question, so we need definitions, we don't have that. And wine quality concerns all developed societies. First, the Minions, they started to classify wines according to their quality, because the high quality wines, they have always a great input in the economy and the society. And this is the case of Burgundy. So today, when we are talking about wine oxidative stability, that concerns all great wines, and especially the Burgundian ones, because they're high-value wines. And as we know, wine is not like a simple food matrix. Wine, for me and for my collaborators, it's a live organism that evolves during aging. So wine during aging is going to evolve and goes through different organoleptic profiles, and until it's achieved its optimum of quality, we denominate the peak of maturation is the bouquet of the reduction. Unfortunately, all wines cannot go through this optimum of maturity, and they go through oxidation directly. So they lose typicality very soon, and they develop some oxidative flavors. Oxidation is very related to the wine resistance against aging, and we know today that great wines that are produced in different regions of the world have a stronger resistance toward oxygen. So oxygen is considered an enemy of white wines until now, because it involves some cascade of chemical mechanisms that can produce reactive oxygen species, and these oxygen species can directly oxidize the main antioxidants that we all know in wine, that are polyphenols. In terms of preservatives, we need to add some antioxidants like sulfites, and wine has also some natural antioxidants like peptides or thiols that they can really prevent oxidation. So the main concern is why we have some wines that can really age for years and others that cannot. In terms to respond to that question, we have developed a multi-crossed approach where we combined 3 different axes. The basis of our work is the use of high-resolution or ultra-high-resolution mass spectrometry, and we apply some metabolomic approach in order to identify known and unknown metabolites that are implicated in wine oxidation evolution. At the second time, we need to understand the mechanism, so we are chemists and we love fundamental reactional chemistry, and based in our knowledge concerning the chemical initiation reactions based on the Fenton reaction, we supervised the radical chemistry by two main tools, the DPPH assay and some EPR approaches, electronic paramagnetic resonance approaches. The third axis is the most important for us because it concerns the identification of molecular markers, and for that we need some old school chemical approaches like LCMS or NMR identification of and structural characterization of some markers. For this project, the first preliminary data before we built our hypothesis was based in FTIR-MS metabolomic approach, where we have analyzed wines from Burgundy from different vintages and different terroirs, appellations. And we understood, after statistical analysis, that we have two main groups that were characterized as a good example. That means wines with no oxidation flavors, the green one, and the oxidized one, that they had a very distinctive molecular fingerprint. So we can see on the right part of the graph that the diversity of chemical markers that are related to stability, the green one, samples, are different to that are implicated to the oxidized ones. By profiling these markers, we understood that thiol, sulfur, and nitrogen-containing compounds that are presented in green and in red color are compounds that are really implicated to wine stability. By applying MS-MS analysis, we could identify some families from each group, and concerning the oxidative stability markers, we could identify three main groups that implied sulfonated polyphenols, sulfonated aldehydes, and sulfonated peptides or amino acids. So we understood for the first time that sulfonation reactions in wine matrix during aging is an important stabilizer for the wine ageability. And what's the point? The point is that sulfites are coming from yeast, but they mainly comes from the winemaking, because the winemakers, they add sulfites to stabilize the wine matrix. We add some free sulfites, and only the 1/3 of the added sulfites are remaining in the free form, were very active, and the major part of the sulfites are bound to known or unknown compounds. The strength of the binding is depending on the chemical characteristics of the binders, and we have a classification provided from the literature that can give more or less an idea about the strength of the binding of known compounds today. So until now, we consider that this bind form of sulfite is a waste. That means that the winemakers, they need to add more and more to increase the percentage of the free fraction. But our results showed that sulfonated compounds are important stabilizer for the wine oxidative stability. That's why we built a hypothesis where we could need to understand how this bind, until now waste molecules, can really stabilize our matrix in terms to reduce the quantity of the added sulfites during winemaking. For that reason, we started with the synthesis of some binders. We examined the antioxidant activity of these binders, and we classified them according to the reaction rate. And of course, we needed the proof that it can be related to general wine oxidative stability expression. So the first thing that Sophia did during her PhD was to monitor the free and the bound form online directly. And this is completely new from the science today in terms of my knowledge, because until now it was impossible to monitor in wine pH condition, which is acidic, this kind of equilibrium between the free and the bound form of sulfites. So today we can report for the first time a direct analysis of sulfones in wine matrix where we can quantify the free and the bound form. Based on that method, we could follow the binding reactions during aging. So in terms of winemaking, we separate it in two parts. The first part is the aging during the winemaking, the barrel aging. And the second part is the aging in the bottle. So we can clearly understand that, especially during barrel aging, where the sulfites are in excess in presence of oxygen, because we have an oxygen intake which is regular. We have especially some aldehydes, ketonic acids, that can bind very fast and directly the sulfites. And we have some thiol-containing compounds, especially peptides or amino acid, that comes after. That's also completely new, because until now, we could really trace the binding reactivity of known and unknown sulfur-containing compounds. And in terms of the bottle aging, we could understood that the today known and really identified as strong binders, like aldehydes or ketones, are not really strong, and they can, their kinetic decrease, their concentration decreases during time. That means that during aging, we have a transformation of the equilibrium between the bind forms and the free forms, and that means that we have some other compounds unknown until today that can really be part of the sink of the bound sulfites. In terms of the reactivity and the antioxidant capacity, we did some measurements of the antioxidant capacity of the free and the bound, newly identified bound forms, and we have the proof that the binding ketonic acids, aldehydes, and some phenolic compounds are more antioxidants than the free ones. That means that the bound, we have the conclusion that the bound forms can be considered as new antioxidants. In order to get a better idea about the real impact of one or a pool of molecules in wine matrix, we supplemented wines with the binding forms, and we clearly could see that when these binding forms are present in very complex wine matrix, we had a direct response in terms of the oxidative stability of the wines. That means that we have the direct proof that these compounds are really implicated in wine's oxidative stability. In order to get on with the anti-radical also activity, and we are working on identifying the mechanism, the chemical mechanism, which is implicated on that, we did some EPR spectroscopy experiments where we can monitor the kinetics of the formation of some radicals after chemical initiation of the oxidation, and we understood that for one more time, when these binding forms are present in the wine becomes more antioxidant. To conclude, today we proposed an NMR-based direct monitoring of sulfonation reaction. That's completely new for our science. And we have the proof that sulfonation reactions are very important in wine oxidative stability. That means we can nowadays consider the binding sulfites as as a potential antioxidant fraction of the wine matrix. And for the first time that we have reported some other compounds, sulfur-containing and nitrogen-containing compounds like peptides or amino acids are potential binders and that can give better antioxidant stability in wines. So now it's the second year of the PhD, and we try also, have only 30 seconds, we try to develop a new NMR-based method in order to estimate the available, that we are going to call sulfites in wines. That means the fraction of the sulfites bind on free one, which has an antioxidant role in wine enjoyability. And of course, in order to diminish the input of sulfites in wines, we are trying and we're going to test some other natural extracts from plants or essentially from plants that can become or can be considered as natural antioxidant in order to complete the picture of our approach. I'm hoping that I keep in time. Thank you for your attention and continue to enjoy.